Good afternoon, everyone, and welcome to the annual Faculty Research Celebration. For those of you who have not met me yet, I'm Katherine Newman, and I have the pleasure of being the interim chancellor of this distinguished university. And this is a fantastic event that I'm very pleased to be here for. Research has been the core of my being for the past 35 years or so, and that up to and including my current job. I think it's the essence of what we try to do in a research university of this kind. And today we have the opportunity to highlight and celebrate the accomplishments of our extraordinary faculty and their contributions in the realms of research, scholarship, innovation, and especially to thank them for their mentorship and support of these extraordinary students who will be the kernel of research for the future. Since 2006, research at the University of Massachusetts has grown exponentially. It's up 170% from 26 million in 2006 to about 70 million in 2017, and that is an incredible track record, which we hope to see continue its ascent. Research in all kinds of areas, from community-based research to the frontiers of basic research, including, and this is just a modest list, developmental psychology and brain science, green chemistry, climate change and its impact on coastal communities. You've heard a lot about that in the last week or so. Aquaculture, gerontology, population and exercise health, sustainability, quantum information and quantum computing. We have Bala here today. That's one of his claims to fame. Remote sensing, environmental chemistry, early childhood and STEM education, and leading efforts on inclusion through research and training at both the national and international levels, just to name a few areas in which UMass Boston has tremendous expertise. The theme of this year's celebration is the teaching soul, research and mentoring, and today's program is going to showcase two faculty members who are working with students who are supported by the UMass Dana-Farber Harvard Cancer Center, the U54 partnership, something I know the university is extraordinarily and justifiably proud of. Since 2002, this partnership has collaborated on research regarding cancer health disparities in disenfranchised populations. It has enabled UMass Boston and the Dana-Farber Harvard Cancer Center to share scientific data and technology that increases research and training opportunities, especially for minority students, and to ensure the sustainability for future research projects and grants. Through research programs in the areas of basic science, population science, student and investigator training, and community outreach, the partnership serves to improve research, training, and outreach opportunities for underrepresented minority students, fellows, and scientists, and to develop information and resources toward closing that cancer health disparities gap. The ultimate goal is to have an impact on society and to educate all of those who are affected by cancer. Since its inception in 2002, the partnership, and this is extraordinary, so buckle your seatbelts here, has trained over 250 undergraduate students. It has increased research funding by over 20%. It has supported 23 innovative research projects across several areas of basic biomedical and population science, and most of all, has resulted in the establishment and the management of our extraordinary Center for Personalized Cancer Therapy here at UMass Boston, and that is something of which we are very proud. It has also provided the opportunity to participate or collaborate in community-based events or health-focused activities. The faculty presenters that we have here today are beneficiaries of this partnership, and we celebrate their commitment to research across their respective disciplines and grateful for their mentorship. We also, of course, celebrate the mentees themselves who have benefited enormously, I know they would agree, from the relationships they have with their faculty, and I know their lives have been affected for the better because they have this enormous individual and career guidance and exposure to the public policy questions that affect health disparities. We know that employers today greatly value this kind of solid academic foundation, and this university offers students like the presenters you're going to hear from today the opportunity to obtain both through the Beacon Student Success Fellowship Program, which supports unpaid experiential learning opportunities for students over the summer, including internships, service learning, civic engagement projects, mentored research projects, and much, much more. We're going to listen to two faculty members talk about how their students inspired them today, and we'll hear from the student mentees about the ways in which they've begun to map out their futures thanks to the support of their faculty. In a minute, my colleague and Provost Emily Mcdermott will formally introduce our presenters, Professor Changmei Kai from the Center for Personalized Cancer Therapy and the Biology Department, and his mentee, Jude Owiridu, who studied biology and is a 2018 graduate of UMass Boston, and Professor Kelly Siegfried from biology and her mentee, Anulawap Ajo, also a biology major, who will be graduating next May. As we prepare to get underway this afternoon, let me remind you this is a celebration of research and relationships, and just how powerful the two are when they're put together. These relationships from both the faculty and student perspectives enhance our whole community and mark the Commonwealth with excellence in research. Thank you all for joining us, and please now welcome my colleague, Provost and Vice Chancellor for Academic Affairs, Emily Mcdermott. Emily?
Good afternoon. So I'm not going to say much, but bring these people up. I just have to say as a footnote at the beginning that because I'm a classicist myself, I have a special relationship with the word mentor, because Mentor was a character from the Odyssey who was a great teacher, and that's where the word is coined from. And as a result, I have a conflicted relationship with the word mentee, because that would be like a Freddie or a Joan E. It's not really a proper coinage of a word, but I don't have any problem with the actual phenomenon of mentors and the students that they provide mentoring to. And I would like to therefore first bring up Changmei Kai of the Department of Personalized Cancer Therapy in the Department of Biology to talk about the project that he undertook with Jude Owiridu, a biology student who graduated this past May, on the topic of somatic mutations in prostate cancer that have included the particularly vulnerable demographic of African Caribbean men. Thanks.
All right, so good afternoon, everyone. Thank you for coming. It's been a great honor to present some of study we have done, with the support of a U54 partnership grant. My name is Changmei Kai. I'm the assistant professor in Department of Biology, and I'm also work for the Center for Personalized Cancer Therapy. So this project is a collaboration with my group and Dr. Franklin Huang's lab in Dana-Farber and Dr. Alexandra Schultz's lab in MGH. So first, my lab is focusing on studying prostate cancer. So what is prostate and prostate cancer? Prostate is a small gland size of a walnut in men that function to make semen, which I think is, the prostate is here, so which carries sperm from the testicles. Prostate cancer is well known as a hormone-driven cancer. Its initiation and development requires the activity of androgen and androgen receptor. So in USA, prostate cancer is one of the leading causes of cancer-related death. It's second in men after the lung cancer, and it's third overall after breast and lung cancer. So for 2018, the estimated new cases for prostate cancer is about 164,000, which is about 10% of all new cancer cancer cases. And the estimated death in 2018 is about 29,400, which is about 5% of all cancer deaths. So if you look at the incidence of prostate cancer for 2015, or it's about as similar as 40 years ago in 1970s. Although there's increase in 1990s, I think this is because of introduction of PSA screening. However, if you look at the death rate, it's actually declined by year. So now we have only about a half of the death rate compared to 1970s. And for the 5-year survival rate, it's actually pretty good now. So for 2015, we have about 98% of 5-year overall survival rate. This is in comparison with 1970s, we have only about a 60% of 5-year overall survival rate. So that sounds pretty good for prostate cancer overall, but there's a significant racial disparities within prostate cancer area. The incidence, morbidity, mortality rate, they vary substantially by race and by ethnicity. In particular, African American men have the highest rate of prostate cancer and worst outcome in response to prostate cancer treatments. So here, we'll just show data published recently by Dr. Timothy Rebeck. He's also one of my collaborators as well. So this is measuring the ratio of African American versus European Americans. So if it's 1, that means no disparity. So basically, where's the over here? So that's the baseline. So if it's over 2, that means it's a significant disparity. You can see, particularly for the younger ages groups, there's a much substantial disparity presented for African American men versus European Americans. Here is a molecular study recently conducted by Leva Gary's lab in Dana-Farber, and one of our collaborators, Dr. Franklin Huang, is the first author of this research. So what they did is to sequencing about 100 African American prostate cancer patient samples, and then compare that with the European American samples. So what they found is there's some genes, their mutation rate is substantially increased in African Americans. So we have listed these mutations here, including SPOP, ERF, and FOXY1. So in their study, they primarily focus on the ERF study, and I think in our project, we actually focusing on the FOXY1. So there's even more serious kind of racial disparity in Caribbean region. So the African Caribbean men have the highest prostate cancer mortality rate in the world. You can see this is the red bar represents. So, and unfortunately, no studies on prostate cancer mutations have included this demographic. So that's formed our kind of a foundation for our U54 project. We propose 2 aims. The first aim is on genomic analysis of prostate cancer in African Caribbean men. In this project, we particularly focus on African Jamaicans. And the second aim is functional characterization of identified mutations. In this aim, we're particularly focusing on the FOXY1 gene, which is well-known. It's a well-known critical gene that functions to regulate the action of androgen. So the prostate cancer development is really requires activity of this gene. So here we show the flow of our project design. The first step, we will collect about 100 prostate cancer samples from Jamaica men. In this case, we used the Gleason 7 and higher prostate cancer specimens. That means more aggressive cancers. And this is done in Jamaica by Dr. Jennifer Alexandra, and also administrated and coordinated by Dr. Alexandra Shields' group. So once the samples are collected, it will ship to Dana-Farber for DNA extraction. And then the DNA will ship to UMass Boston, the CPCT Genomics Core, which is led by Dr. Jill Makowska. And then we're going to perform so-called targeted sequencing to examine the mutations on about 200 tumor-related genes. And then that data, the results of sequencing will be transferred to Broad Institute. Dr. Franklin Huang's group will be conducting bioinformatic analysis to find the specific genes that may be related to the African Caribbean man. And hopefully we can identify certain specific mutations in FOXP1, and then we're going to conduct functional analysis and molecular analysis on this gene in our lab. So that's the whole process of the project. And so far, it's not finished yet, but we already finished collecting samples. So the 100 prostate cancer specimens are ready to ship, and then we hope we can get that as quick as possible and then conducting the DNA extraction. But in the same time, we already started analysis on the FOXP1 genes based on the paper I just mentioned by Dr. Franklin Huang. So we have some very interesting results so far, and we're ready to submit a paper for summarizing these results. So this project has a very significant impact on the research, teaching, and the training. So far we have 2 grant proposals that have been just submitted a couple of days ago to Department of Defense Prostate Cancer Research Program to search for external funding. We actually submitted one last year. We didn't get it funded. And we also have one manuscript is under preparation. We hope we can submit it by the end of this year. And for training, I think this project provides a good opportunity for 2 young investigators. One is my former postdoc, Dr. Shuai Gao. I think he's over there. He was actually recently promoted to research assistant professor of CPCT. And also the Jude Awardu, he's an undergraduate student. He has been working in my lab for over 3 years. I think he will talk a bit more about his experience of this project. And also for the UMB and the Dana-Farber community, we actually also provide a workshop on the cancer genomics and the disparities for particularly undergraduate students. We have about 20-30 undergraduate students participate in this event. I think they have learned a lot from the experts in this area. So I think that's pretty much it for my part. I'm gonna give this to Jude, and he will talk about his experience.
All right, so thank you, Dr. Kai. So once again, my name is Jude Awardu, and I'm here to talk about how the research opportunities and fund opportunities I've had here at UMass kickstarted my research career and helped me get to where I am. So first off, my journey from Ghana to becoming I'm currently on the track to becoming a physician scientist. That is my ultimate goal. And here on the left, that is 4 years ago before I came here to the US from Ghana. And the right 2 pictures are earlier, a couple months ago in 2018 when I graduated and successfully did a poster presentation of my honors thesis. So a little bit about me. I immigrated from Ghana to the US specifically to Boston actually in 2014, and then began studying here at UMass under a bio degree in 2015, the fall of 2015. And in the summer of 2016, I applied for the U54 UMass Boston Dana-Farber Harvard Cancer Center Cancer Partnership, which aims to gear people in minority groups into the world of research and scientific research. And I was successfully accepted into the program, and this is how I began my mentorship under Dr. Kai. So currently, I conduct prostate cancer translational research, which is focused on understanding the molecular mechanisms between molecular mechanisms that govern the signaling and regulation of the androgen receptor in the development of prostate cancer. in the hopes of or in the aim of providing or developing treatments which can effectively target this disease. So I'm here to talk a little bit about the funding opportunities I've had so far these past 2 years whilst undertaking research. The first I would like to highlight is the UMass Boston Dana-Farber Harvard Cancer Center E54 program. This program was key and pivotal in kickstarting my research career, and I would like to shout out and give a special shout out to Ms. Deidre writing on the right, who was key in actually helping me realize this program, finding out about this program, and also under her guardianship for the past 2 years. I would say there's no better person who I would have had as a mentor. And I also like to highlight the Sanofi Genzyme funding award, which I applied for and was accepted into twice, and the National Cancer Institute grant, which is affiliated with the U54 program. Also, I would like to talk about this networking event, this regional conference, national conference we have. It's called the Annual Biomedical Research Conference for Minority Students. I attended that twice, and that gave me the opportunity to network with top professionals in the biomedical field and better understand where I want to go and how to get there. So, talking a little bit about my accomplishments, over the past 2 years, I've been able to present at a number of conferences, one being the National England Science Symposium, and the other being the Abercamps, which I just talked about in 2016 in Tampa, Florida, and in 2017 in Phoenix, Arizona. In early 2017, we successfully published a paper on androgen receptor mutation in the Oncotargets journal, and currently we submitted a proposal for a paper which is currently being reviewed under here. And finally, I want to talk about my honors thesis, which I submitted in June of 2018, and I'm very proud of that, since it's a direct result of all my work in the lab. So talking a little bit about my future, a couple of months ago, Ms. Deidre asked us to kind of plan out our life, like a life on a piece of paper, a big piece of paper, and this is what I came up with. So we're currently here. Sorry, there's no pointer, but I'm currently graduated, on my way to applying to an MD PhD program, and hopefully getting to the position of a physician scientist where I can go back to my home country and to kickstart or help develop the medical and healthcare system back home. So lastly, I would like to acknowledge the CPCT in general, the Joe Makowska lab, the Kai lab, Charger Patanius lab, and I would like to acknowledge all the members of the Kai Lab, from Dr. Sharagao to Ms. Anna Beshednova. And one thing I would like to say about my lab is it has a very inclusive culture, one that I enjoy coming to every day. And you can see this by the names underlined from Kevin, myself, Amy, and Antia. We are all minority students in Dr. Kai's lab. And I also like to acknowledge Ms. Alexandra Seals' lab, Dr. Franklin Wang, and our international collaborators, the University Hanshin Hayes Lab from the University of Toronto, and also Dr. Stephen Balk and Eva Corey's lab. Thank you.
Okay, if you'll indulge me one more moment about Greek classics. Some of you may be rusty on the Odyssey. But you might remember that the first 5 books of the Odyssey are devoted to the coming of age of Odysseus's son Telemachus, who has grown up from infancy to the age of about 20 while his father was away fighting in the Trojan War. He had his mom at home, but she had her own problems. And his guidance in growing up came from mentor, the elderly teacher who guides him not only by teaching him, but by leading him through life to the point that he can come of age. We've specialized that word in the English language recently to talk about guidance that brings people through to accomplishment in a specific field. We're talking about research today, that the mentors that we have the privilege of meeting today have helped to guide their mentees through to accomplishments as adults in the world of research. And I think that's just such a wonderful thing for us to be able to say that we are able to do at this university. So our second mentor for today's honoring is Kelly Siegfried, professor of biology, whose lab has provided multiple research opportunities for both undergraduate and graduate students. She has served as guide or mentor to Anu Ajao, a senior student in biology who will graduate at the end of this year. And I'd like to invite Kelly to come forward and Anu to talk about their project on understanding the genetic regulation of germ cell development as it pertains to infertility and tumorigenesis.
Thank you for having me. I also am very pleased to be here and honored to be invited to speak at this event to represent the faculty research. I'm also in the biology department. I'm an assistant professor there. And my lab is doing research that pertains to how the testis develop and how it maintains as kind of a model to look at how tissues are maintained through our lives. So I'm going to do my acknowledgments first, and then Anu will speak a little bit later. So my lab currently, I'm mentoring 3 graduate students in the lab. I've had 2 that graduated previously. And these students are really kind of the major researchers in the lab, and my goal is to guide them and kind of help them shape their projects and their direction of their thesis work. I also have had several undergraduate students in the lab, and my current and past undergraduate students are listed there. And what I tend to do in the lab is pair the students directly with the graduate students so they really get one-on-one, day-to-day mentorship from those graduate students. And I think this gives a benefit, twofold benefit in both directions. You know, one, the undergraduate students get this really one-on-one experience with the graduate students, and also the graduate students start to gain experience on how to be mentors themselves. And this seems to work really well, and then I kind of help shape that whole experience for them. Okay, so into our research projects. One of the questions that we're really interested in my lab is how you continuously maintain your tissues throughout your life. And this is a developmental process that occurs during your entire life. And so as you may know, many tissues and organs replenish their cells constantly during our lives. And there's some examples I've listed here. So for example, blood cells are constantly being turned over and need to be replaced. And this also might need to be ramped up in some instances. For example, if you have an infection and need more immune cells, or if you have an injury and you need more blood cells in general. This also occurs in the skin, where your skin is constantly dying and sloughing off, and those cells need to be replenished. And there's many other examples of that. And we're looking at this idea with respect to the sperm cells. So the sperm are constantly being turned over, and those cells need to be replaced because sperm is continuously produced throughout the life of the organism. And so there must then be some kind of balance that's maintained to constantly replenish your cells that are lost. And this has to be really tightly regulated, because if you are producing too many cells, this can lead to potential tumor formation if you have uncontrolled proliferation of the cells, or if you're not producing enough cells, this can lead to tissue loss. And this latter effect can happen during aging, for example, when your skin starts to get thinner, et cetera. These are due to cells not continuously replacing those loss at an adequate rate. And so we're looking at this process in the testis, and so I'm showing you a picture of a human testis here, and packed within the testis are all these tiny little tubes, and within those tubes are where the sperm are developing. And so within then these tubes, if I can get a pointer, there's the sperm are going through development, and so you have these immature cells that go through development, and these immature cells are capable of dividing to produce more cells. And then once these begin to mature or differentiate, they'll form the sperm, which can then carry out its function, which is to fertilize the egg. And once they go through this maturation and differentiation process, those cells are no longer able to divide, so they need to be replenished by the immature cells. And again, this process needs to be tightly regulated so that you're producing the correct number of cells, not too many or not too few cells in the end. And so this is then generally one of the processes that we're looking at. So within the testis, as well as other tissues that are replenishing their cells, you have different populations where, for example, the stem cell is an undifferentiated cell that can undergo a cell division to give rise to more stem cells or another undifferentiated cell called the progenitor cell. And those progenitor cells can undergo more divisions to give rise to a predictable number of cells that will then undergo differentiation to make the fully functional and differentiated cell type. So again, if you have too much cell proliferation, if this becomes unregulated, then this can get out of control, and that's partly how tumors will form. And contrary to this, if you can't produce enough cells to replace the cells that are lost or turned over, then you're going to eventually run out of that tissue, and in the case of the testis, this is going to result in sterility. So these are kind of the processes that we're looking at. And the approach that we take to study these processes is using mutant analysis. And we're doing this in a model organism called the zebrafish, which is a small fish that you can often see in pet shops. So the zebrafish model has become a really important model for studying developmental process and also for disease modeling for human diseases. And that's because we can easily manipulate this and do experimentation on this fish in the lab. And importantly, 70% of the fish genome has a counterpart in the human. So 70% of their genes are the same as the human genome. And those genes are carrying out similar functions in both the fish and in humans, and often have similar disease effects when they're mutated. So we're using this approach in zebrafish to understand the process of how sperm cells are continuously replaced throughout life. And the way we do this is essentially, if you're interested in a gene and how it functions, one way to test that is to just delete or mutate that gene. And if the deletion of that gene results in sperm loss, for example, that means that gene is necessary for continued maintenance of the sperm population. And so you can conclude then from this that this gene is necessary for sperm development or maintenance. And so this is the technology that we're using in our lab in the fish. And you can take a couple approaches to this. One, you might have a gene that you see that you're interested in, and you want to study it, and so you go in and you mutate it and ask what happens. And the other approach, which is the approach we're often using in my lab, is that you can generate random mutations throughout the genome of the animal, and then look for defects that are in the process that you're interested in. So for example, we're looking at fish that have sterility defects, and then we can go back and identify the genes that were mutated that give rise to those defects. And this approach is my favorite approach because it's really an unbiased way to look for genes that are functioning in a specific process. So I'm going to talk about some mutants that we're looking at. Well, I'm going to talk about one mutant that we're looking at in the lab, and then Anu is going to talk about the mutant that she's working on as part of her project in the lab. So to introduce these, first, in the normal fish, they have a testis, just like the men in this room. I don't have one. So the testis is populated by the germ cells. And the germ cells are the cells that are going to give rise to the sperm, okay? And so they are, just like in the male testis, you have germ cells that are undifferentiated and giving rise to the sperm. So they're continuously used and continuously replenished. I'm going to talk about a mutant where this process is unregulated, so we have uncontrolled proliferation of the germ cells that results in a testicular tumor. And then Anu will talk about her project that has the opposite effect, where there's not enough sperm being made and the fish becomes sterile. Okay, so I'm going to talk about this project, which is the project of a graduate student in my lab named Qazi Islam, who's doing this as part of his PhD thesis work. So this shows you what the fish look like that have the tumors, if you can see it. So these are sections through a fish, and outlined in these dotted lines are the testes. And you can see a normal fish has these nice small testes, whereas in the mutant fish that we have, they have this gigantic testes tissue that fills up the entire body cavity. So we have this major impact of overgrowth of the testes in this fish. And so Qazi did some work on these mutants to ask what was going on and start to get an idea of how why these fish are getting tumors, and also we're interested in how these tumors might compare to human testicular cancers. And so we did a few experiments, and the summary of these experiments is at the bottom, and that's that Qazi found that based on the fact that these have an increased number of undifferentiated germ cells and increased expression of a gene called SOX2, these appear similar to a testicular cancer type called embryocarcinoma, which is the second most common type of testicular germ cell tumors in humans, and is a highly malignant form of this tumor. So the next thing Qazi wanted to know was what gene is mutated to give rise to this tumor formation in the fish. And so what he did was he collected DNA from the mutant fish and sequenced the genome of those fish. And through analysis of that genome, we found a nice mutation that we think might be causing this effect, which is called SEP78. And so SEP78 is encodes a really interesting protein that's both localized to the centromeres of dividing cells, so it's important for cell divisions, and it's also important for formation of a cellular process on the surface of many cells called cilia. And cilia are important for cell movement, for moving fluids within tissues, and also for communication between cells. So they have a lot of functions. And so the SEP78 gene is localized to this centriole in the cilia, which is important for formation of the cilia as well as the centrosome in dividing cells. And there has been recent research that have seen connections between mutations and genes involved in cilia formation and function and testicular germ cell tumors. So we think that this is a really good candidate mutation for causing the tumors in our fish. So in order to compare our data with the human testicular germ cell tumor and try to develop this as now a human model to study this disease, we partnered with two groups at the Dana-Farber Cancer Center with support from the U54 collaboration. So the groups of Chris Sweeney and Ellie Van Allen are doing genomic analysis on human germ cell tumors. And so they have a collection of sequenced tumors, and we're now looking through that data to see if we can find any indication of SEP78 mutations in human germ cell tumors. And to expand this analysis, we're also working with some of the epidemiologists over there to look at a more exhaustive look at other published or available data sets of testicular germ cell tumors for connections either with SEP78 or other cilia-related genes, and see if we can make some connections between our fish model and the human germ cell tumors. So that work is ongoing, but also using our fish model, now we can explore some more broad questions related to what's the role of the cilia in spermatogenesis? Why do we need cilia in spermatogenesis? And also, why does loss of cilia function lead to germ cell tumor formation? So these are now future avenues that we'll be investigating. So the second mutation that Anu is going to talk about is involved in germ cell maintenance. So this is now a mutation where the fish lose their germ cells and are sterile. And so again, this project is part of Kazi's PhD thesis, and he's working together with Anu on this project, and she's going to tell you about her research.
Thank you, Kelly. As Kelly mentioned, my project is focused on germ cell maintenance in mutant zebrafish, and specifically it is focused on the vacant mutant line. And on this slide, we see cross sections of both normal and mutant zebrafish at different time points. So on the left, there is a cross-section of the normal fish at 2 months old, and we see that they have all the germ cells that Kelly mentioned earlier. Right next to it, we see the cross-section at 2 months old of the mutant zebrafish, and we see that it also have the germ cells that Kelly mentioned, most especially the sperm cells. And on the right, there's the cross-section of both the normal and mutant fish at 6 months old. You can see that the normal fish has the same germ cells as denoted by the shades of purple. And right next to it is the mutant zebrafish at 6 months, but you see that they have the testis tubules empty, and they're supposed to contain the sperm cells, which they've lost because they're unable to replenish and produce more sperm. And we think that the reason for this is a maintenance issue. So at some point they produce the sperm, but they lose it. And that has led to the focus of my research questions, which are, first, if stem cell proliferation is effective in the vacant mutant fish, and second, which gene is responsible for the loss of germ cells in this vacant zebrafish. To answer the first question of if cell proliferation is defective in the zebrafish, I did a cell proliferation assay on normal zebrafish testes, and I found cell proliferation assay just marks the cells that are dividing, which are the germ cells in the zebrafish testes, and they're marked on this slide by the dark cells that the arrows are pointing to. And just to finalize my results, or to identify, to determine if cell proliferation is effective in the mutant fish, I will be repeating this same process on the mutant zebrafish, and what we expect to see if our hypothesis is right, that the cause of the sterility is due to germ cell maintenance, we should see a reduction in the cell proliferation when we compare the mutant zebrafish testes to this image that you're seeing right now. And to answer the second question of which gene is responsible for the sterility in the vacant mutant line, we sequenced the genome of both the normal and mutant zebrafish, and then identified a general region in the genome where the mutation is. And we also found 2 mutations in this specific region that are present in the mutant fish, but not in the normal fish. So this lets us know that either one of the 2 mutations that we identified is the cause of the sterility in the zebrafish, in the mutant zebrafish. And the 2 candidates gene that I'm currently studying in the lab are ADAD1 and SLC34A1A. And although we do not have much information about both genes right now, there's not much research done on either of them, but we do know that they're both present in the human genome, which means that if either one of them is the cause of the sterility in zebrafish, it is likely to be a cause in humans, in a human condition as well. So to determine which of the two genes are the cause of the sterility in zebrafish, we we created additional mutations using CRISPR-Cas9, which is a genetic tool that is used to edit the genome either by adding or deleting sections of the genome. And we've created new mutations for AD81 and SLC34A1A using this technique. So we'll be breeding this the new mutants and just comparing to see if they have the same phenotype as the original fish. And just in summary of my project, in summary of the project that I've been working on in the lab, it is focused on identifying the cause for a specific form of sterility in zebrafish that could potentially give answers to the infertility in humans. And overall, our lab is just focused on using zebrafish as an animal model to understand genetic regulation of testicular cancer and infertility. And results from this can be used to identify or just lead to development of targeted therapies and diagnostic tools. And just in closing, I would like to thank Drs. Deidre Ryden and Dr. Emily Jones, she's not here right now, just for giving me my first opportunity towards research. It was not a wet lab, but it was definitely the first step for me to research. And I just want to thank Dr. Kelly Siegfried for also giving me the opportunity, because we met at a U54 symposium last year, and she was the first person to give me the chance to do research, even though I had no experience other than my classes. And just without her, I don't think I would be able to know as much as I do right now. And just like Jude, the opportunity that I've had in the lab for the past 10 months has definitely influenced my decisions for the future. And initially I was thinking strongly towards an MD degree, but now I really want to do more research and definitely incorporate my research into the kind of medicine whatever medicine I go on into, and perhaps that could be an MD, a dual degree, an MD PhD, or maybe just a master's in science. Thank you.
That was inspiring, and I know in a few years we'll invite these two students back to UMass Boston, and you will do us proud, and maybe you'll join these faculty members if the spirit moves you. We certainly need more outstanding scholars in our laboratories, and it's just an honor to know that you got your start here, and I'm grateful to your faculty mentors for helping you along that pathway. Before closing, I want to acknowledge and thank Joanne Riley and Mary Moser. Where are you? Joanne and Mary? From the Healy Library. And Mark Kenyon, where are you, Mark? He was here before. From the Career Services and the staff from University Events and Ceremonies for organizing this important event. It's really wonderful to have a chance to celebrate with all of you. And now let's go really celebrate at our reception. Please, right through the door. Thanks again.